you for this introduction, and also the organizers for their kind invitation to present our data here. I was asked to talk on astrocytes in respect to noradrenergic regulation of their metabolic and morphological plasticity, and also therapeutic opportunities. So astrocytes, as you may all know, they are non-neuronal cells, they are glial cells, and compared to neurons, they are not electrically excitable, but they can still respond to signaling molecule in the brain through receptors that are, just a second, through receptors that are expressed on their surface. So many of these receptors are actually metabotropic receptors, GPCRs, and their activation leads to increase in secondary messengers such as calcium and cyclic AMP. And through this so-called cytosolic excitability, these cells can regulate many processes, and I would like to point out here that these are actually the main cells, the key cells that are taking care of our brain homeostasis. And we all know when brain homeostasis is not working well, we may actually get disease, and astrocytes have been involved actually in all diseases that are known. They have been linked to those diseases in some way. I will, in this talk, focus on their maintenance of metabolic and water homeostasis. They are very strategically positioned in the brain between blood vessels and the synapse, and they can thus uptake glucose and other metabolites, store them in the form of lipid droplets or glycogen, and then when needed, when neurons are under stress or they have increased activity and need more energy to maintain action potential, astrocytes can deliver these nutrients to neurons. Also, they express many water channels, aquaporin 4 channels, and they are also involved in the regulation of the lymphatic systems that I will talk later. So, what is noradrenergic system? The system in our brain originates in the nucleus locus ceruleus, which is in the brainstem, and from this area it projects axons throughout the brain, and when the system is excited, noradrenaline is released throughout the brain and activates cells. And it has been shown recently that actually in the last decade that astrocytes are the main effector cells of the system because they are rich in the adrenergic receptors, and when you excite the system you can see global excitation, calcium excitation in the cortex area, particularly here in this image. So the system controls brain metabolism, memory and learning, attention, sleep-wake cycle, plasticity, stress response, and all this has been also linked to the function of astrocytes, further showing us that actually these cells are very much linked to the locus ceruleus noradrenergic activation and modulation of neurons. So actually astrocytes are the first target, and then downstream, of course, this affects the neuronal function. So what actually noradrenaline does in astrocytes, we were interested in that. I'll just briefly show you. Then when you apply noradrenaline and measure with the fluorophore AMDI, the calcium signals, you can see fast increase, and then which is transient with oscillatory manner. But when you measure cyclic AMP here in red, and actually express sensors for cyclic AMP like pink flamingo, in astrocytes you can measure in the same cell a slower response, which actually is persistent, remains high. The level of cyclic AMP remains high throughout exposure to noradrenaline, and that shows us actually that the response is the temporal dynamic of the response is for cyclic AMP and calcium different. Many studies have been done for calcium, but we were also interested in our studies, which I don't have today time to talk on especially on cyclic AMP. And during such a study, when we were measuring cyclic AMP in astrocytes with another type of sensor, we noticed that adrenergic activation of astrocytes causes morphological change in astrocytes. Actually, those cells start to shrink. They start to produce processes, so they become not more stellate, so not a fibroblast-like type of morphology, changes actually to stellate morphology, and this occurs also in situ if you stimulate astrocytes. So I already mentioned aquaporin channels in the beginning, and these channels have been shown to be very important in astrocytes for the regulation of the lymphatic system. This is a system that keeps our brain clean, actually clears the waste from the brain, and astrocytes, as I said, they are involved in that, but aquaporin channels could be also toxic for the brain, because these cells are during traumatic injury actually the first cell that starts swelling. So these are the cells that cause cellular edema in the brain, and this is the first stage of brain edema. Then we have 3 other stages that lead to hemorrhage also in the brain. So our idea was whether we can control this swelling by increasing cyclic AMP in astrocytes. So we first did some in vitro tests. We stimulated astrocytes with hypotonic solution. You can see that they start to swell. This is the volume change. But when you apply adrenaline or isoprenaline, which activates beta-adrenergic receptors, you can reduce the swelling of astrocytes. And then we moved to the in vivo system. We introduced swelling of astrocytes in an animal model that expresses GFP, so astrocytes are labeled in the brain. This is transgenic animal, and you can actually follow the increase in volume in those cells by application of intraperitoneal injection of water in mice. And within 1 hour of this application of water, you can measure swelling. This is the blue graph here, swelling of astrocytes. And we collaborated with the laboratory that has this animal, and we found out that actually when we apply adrenaline to the brain, we can prevent swelling of astrocytes. Then we moved to a more pathological setting, and we use another model, rat model of concussion spinal cord injury. When you apply injury to the spinal cord, again, you can see within 5 minutes of the concussion, we can see swelling of neighboring astrocytes. That means that cellular edema started. Again, also in these experiments, when we applied topically on the spinal cord injury adrenaline, we could prevent or highly actually reduce swelling of astrocytes. We labeled cells here with these dyes, and these are different 3 types of analyses that were made on astrocytes to confirm these results. So then our question was, of course, what is the mechanism behind? We know from before that when you apply hypotonic medium, you get calcium oscillations, which are actually were linked in the past to activation of mechanosensitive channels on the surface of astrocytes, which are actually enable calcium to enter the cell. And when we applied isoprenaline or adrenaline to cells, you can see that there is a reduction in these oscillations. And probably this mechanism is actually involved also in the reduction of volume of astrocytes, which we are still trying to evaluate into more detail. So from this we concluded that with activation of adrenergic receptors, or at least even if we don't use adrenergic receptors, some other receptors that are expressed, this is our idea, only on astrocytes, we could actually increase cyclic AMP specifically in astrocytes, and maybe in the future use this mechanism also as a therapy to treat cellular edema and prevent further edema formation. So now I will switch to next part of my talk, to metabolism. I already mentioned noradrenergic system regulates metabolism. And just briefly, when you activate the system, or when the brain is under stress, because noradrenergic system is a major stress response system in the brain, you get, you can observe in astrocytes glucose uptake, glycogen degradation, and then in the process of aerobic glycolysis, glucose is then transformed into lactate, despite normal oxygen levels, and lactate is believed to be shuttled to neurons through transporters, MCT2 transporters, and some other channels, and used as oxidative fuel. And this is very important. Some tests showed on animal models that these pathways is very important for cognition and neuronal function. So you can easily measure this also in cultured astrocytes. When you apply noradrenaline, you trigger glucose increase, indicating glucose uptake from the extracellular space, and aerobic glycolysis. When you measure lactate, you can see increase in aerobic glycolysis and lactate production. So what we did, we started to collaborate with Boris Rogel, because he had he works on ALS, and he has actually made plasmids that carry TDP-43, which is the protein that is involved in the pathology of ALS and FTD. I will not go into detail what these diseases are, because I think everybody heard during this conference, which is ALS oriented, also partly a lot about the disease. But I would like to say that in, I don't know, last decade, scientists started to believe that maybe glial cells, in particularly astrocytes, could be the drivers of neuropathology in TDP-43 proteinopathies, because this protein, which forms in when the disease occurs, inclusions outside, usually it's inside the nucleus, but then it forms inclusions outside the nucleus, which are toxic to motor neurons. But this has been shown that these inclusions can be found also in astrocytes, and they actually are sufficient to cause motor neuron death in animal models. So even if you don't find inclusions in neurons, astrocytes can actually, with inclusions, be toxic for neurons. So what we did, we introduced TDP-43 wild-type protein that was tagged to red fluorescent protein, and truncated form, which is involved in pathology in ALS, also labeled with red fluorescent protein, into our in vitro model of astrocytes. And you see TDP-43 is present in wild types in the nucleus, while in the truncated form, the truncated form is outside the nucleus. We then checked whether these cells are under stress, and we labeled cells with the lipid droplet marker. This is a stress response marker, and indeed in cells with TDP-43 inclusions, we could see increase in accumulation of lipid droplets, also perhaps suggesting that these cells are moving from glucose metabolism to lipid metabolism. When we looked at the noradrenergic system, we could see that there is a reduction in calcium and cyclic AMP signaling in cells with TDP-43 inclusions. Moreover, we also have seen that there is reduction in the expression level of beta adrenergic receptors, therefore probably cyclic AMP signals, but not in alpha-1 adrenergic receptors, which are linked to calcium signals. So, to our surprise, we also noticed that when we apply noradrenaline, we can trigger lactate production, so aerobic facilitation of aerobic glycolysis. But that was not different between cells with the wild-type TDP-43 and the truncated form. To our surprise, even more cells were responsive to noradrenaline treatment with lactate production, suggesting that even more lactate is produced and perhaps released and supports neurons. But then we looked at the level of expression of MCT transporters in astrocytes. These are lactate transporters needed for lactate to leave astrocytes and then later on enter neurons. And we could see that there is reduction of expression of lactate transporters, suggesting that actually, although we have downregulation of cyclic AMP and calcium levels in astrocytes upon noradrenergic activation, this does not affect actually lactate production. If you look at the whole cell population, maybe even more lactate is produced in cells. But lactase perhaps stays inside or is trapped inside, not efficiently leaves the astrocytes, and therefore this would mean that we have reduced neuronal metabolic support, what could actually contribute to pathology. And thus, astrocytes may represent in the future a novel drug target in both cases. At the end, I would like to acknowledge the co-workers and also at the Institute of Pathophysiology in Ljubljana that are listed here, that primarily worked on these projects, but also all the others that contributed. And at the end, also, I would like to thank the funding agency. Thanks.